Somewhere inside every one of us, there's a story older than any civilization, older than any language, older than any border ever drawn on any map. It is written not in ink, but in the twisted microscopic architecture of our DNA, a molecule so ancient and so faithful that it has been copying itself generation after generation for hundreds of thousands of years. And for the first time in human history, scientists have developed tools powerful enough to read that story in full, not just in fragments, not just for one population or one continent, but for every major human group on Earth, tracing their genetic ancestry through space and through time, watching mixed with what crises they survived and how the modern world came to be assembled from those millions of overlapping journeys. What they found rewrites much of what we thought we knew. Most populations fit into the story neatly enough: African, European, East Asian, South Asian, Native American, each with their own branching paths, their African continent that began nearly 200,000 years ago. The picture that emerges is complex, fluid, and deeply interconnected. There are no pure lineages anywhere. There never were. Every population on Earth is, to some degree, a mosaic, a layered accumulation of people who moved and settled and merged and moved again, generation after generation, leaving their genetic signatures behind like handprints pressed into the ancient rock of our chromosomes. But there is one population that does not fit cleanly into this picture. One group whose DNA sits in a category almost entirely its own, neither fully European, nor fully Middle Eastern, nor anything else that the standard continental labels can capture. A group that forms its own distinct genetic cluster in every major study ever conducted, separated from their geographic neighbors by centuries of enforced isolation, religious endogamy, and periodic persecution, and by a biological event so catastrophic and so precise that the entire modern global community descends from just a few hundred founding individuals who lived in medieval Europe roughly 7 centuries ago. That group is the Ashkenazi Jewish people, and understanding why their DNA stands apart the way it does tells us something profound, not just about them, but about the nature of genetic identity, the fragility of human populations, and the extraordinary power that history holds over biology. To understand what makes Ashkenazi genetics so unusual, you first have to understand how the broader science of human DNA mapping actually works, and how dramatically that science has advanced in just the past few years. For decades, geneticists described human populations using broad continental categories. You were European, or African, or East Asian, or South Asian. These labels were convenient, and they captured real patterns in the data. Human beings who share ancestors tend to share DNA variants, and those shared variants cluster together in ways that roughly mirror geography. That much is true. But convenience is not the same as accuracy, and simplicity is not the same as truth. The categories always missed something enormous: the ancient migrations, the admixture events, the ghost populations, who left traces in modern genomes before vanishing entirely from the fossil record. The labels gave us a map, but a map, as anyone who has ever walked in wilderness knows, is never the territory itself. In 2025, a landmark study published in the journal Science changed the conversation in ways researchers are still absorbing. A team at the University of Michigan, led by Michael Grundler, Jonathan Terhorst, and Gideon Bradford, developed a method that does something no genetic tool had ever managed at this scale. Instead of assigning people to fixed geographic categories, it traces the actual locations of shared ancestors through time, watching individual DNA segments travel across the landscape over thousands of years, mapping not just what you are genetically, but where your genes have physically been. The method relies on something called an ancestral recombination graph, a mathematical structure that encodes the entire genealogical history of a genome at every position simultaneously, and applies it to sequences sampled from populations across Europe, Asia, and Africa. What came back was extraordinary. The method accurately recovered every major population movement the researchers tested it against. It traced the expansion of early farmers out of the Fertile Crescent into Europe and beyond. It mapped the great steppe migrations that reshaped European genetics during the Bronze Age. And most fundamentally, it confirmed something that simpler analysis had always implied but never demonstrated so cleanly: that every human alive today, regardless of where their recent ancestors came from, carries African DNA when you trace back far enough. Around 200,000 years before the present, all of our genetic histories converge on a single continent. The notion of a non-African lineage is not just a social construction. It is biologically incoherent when viewed at sufficient timescale. We are all, in the deepest and most literal sense, children of Africa. But that same study also made clear that the history between that distant origin and the present day is anything but a simple linear divergence. Populations separated for tens of thousands of years do accumulate real genetic differences, real distinctive variants, real divergent frequencies at thousands of positions across the genome. And ancient DNA has shown us repeatedly that the people living in a region today are often not the direct descendants of the people who lived there thousands of years before. Europe is the most studied example. The people we call Europeans today are a layered mixture of ancient hunter-gatherers, early farmers who arrived from Anatolia roughly 9,000 years ago, and steppe pastoralists who swept in from the east during the Bronze Age, around 4,000 to 5,000 years ago. Not one of those three contributing streams survived intact. They blended. They were replaced in some regions and absorbed in others, and the resulting mixture is what we now call European, a thus category that carries within it the traces of migrations its modern members know almost nothing about. The same is true for virtually every population on Earth. Advances in the study of ancient DNA have revealed a profound lack of genetic continuity within geographic regions, a finding that makes the very notion of stable, bounded ancestral populations far less tenable than most people assume. Around the same time, another major advance arrived in the form of a tool called Orchestra, published in Nature Communications in May of 2025. The name is an acronym, Optimal Recombination of Haplotypes to Establish Segmentation of a Target from Reference Ancestries, and it was trained on the genomic data of over 10,000 individuals drawn from 35 worldwide populations. Its specialty is what geneticists call local ancestry inference, the ability to examine a specific stretch of a person's DNA and determine, with fine precision, which ancestral population that particular segment came from. Earlier tools could do this in broad strokes, but Orchestra operates at a level of resolution the field had not previously achieved. It can detect trace ancestries, the genetic contribution of a great-grandparent, a single immigrant ancestor, a community that merged briefly with another centuries ago, that older methods would have smoothed over or missed entirely. When Orchestra was applied to Latin America, the results were a remarkable confirmation of the historical record. In the Guianas, it detected elevated Indian ancestry consistent with the British colonial indenture system, which brought tens of thousands of South Asian laborers to the Caribbean and South. But it was when Orchestra turned to the question of Ashkenazi Jewish origins, one of the most contested questions in population genetics, that the findings added the most to a decades-long scientific conversation. The Ashkenazi Jewish people are, genetically speaking, unlike almost any other population on Earth. That is not rhetorical overstatement. In every major genomic study conducted over the past 20 years, from early SNP chip analyses to the most recent whole genome sequencing projects involving thousands of individuals, Ashkenazi Jews form their own distinct genetic cluster. When researchers plot global genetic variation using dimensionality reduction techniques, most populations arrange themselves roughly along geographic lines. Europeans in one region of the plot, East Asians in another, Africans in a third, South Asians somewhere in between. Ashkenazi Jews appear between Europeans and Middle Eastern populations, clearly related to both but not fully belonging to either. Their internal genetic similarities, the degree to which any two randomly chosen Ashkenazi individuals resemble each other genetically, is far greater than their similarity to the European populations they lived alongside for 1,000 years. It is a signature that appears instantly, unmistakably, in every analysis ever run on this data. The history behind that signature begins with migration. The Ashkenazi Jewish community descends from Jews who moved into Central and Eastern Europe during the early centuries of the Common Era and the medieval period, establishing communities across what is now Germany, France, Poland, Hungary, Ukraine, and Russia. They brought with them a genetic heritage rooted in the Middle East, specifically in the Levant, the ancient homeland of the Israelite people, and they arrived in Europe carrying that ancestry largely intact. What happened next is what makes their genetic story so unusual. For roughly 1,000 years, and in many places much longer, these communities lived in close geographic proximity to majority European populations while remaining largely endogamous. They married within the community. Religious law encouraged this. Social exclusion enforced it. Persecution reinforced it further. Jews were excluded from guilds, barred from land ownership, confined to specific urban neighborhoods, expelled wholesale from entire countries, and subjected to cycles of violence that drove communities inward on themselves for protection. The biological result of this sustained separation was a slow but continuous accumulation of genetic distinctiveness. Generation after generation, the Ashkenazi gene pool remained its own closed circuit, diverging steadily from the European population surrounding it, growing more internally homogeneous with every passing century. Then came the bottleneck. Genetic evidence converges on a period roughly 600 to 800 years ago, somewhere in the late medieval period, when the effective size of the Ashkenazi founding population was reduced to a number that strains credibility when you first encounter it. The most careful estimates, derived from the lengths of shared DNA segments between living Ashkenazi individuals, put the founding effective population at somewhere between 250 and 420 individuals. Not total community members, not all the Jews living in Europe at that time. Effective founders, the number of individuals who actually contributed meaningfully to the gene pool from which all modern Ashkenazi Jews eventually descend, a number that would fit comfortably inside a single apartment building. The implications of that single fact ripple outward in every direction. The approximately 10 million Ashkenazi Jews alive today, spread across Israel, the United States, Argentina, the United Kingdom, and dozens of other countries around the world, are all distant genetic cousins of one another in a sense that goes far beyond metaphor. Studies have calculated that the degree of shared DNA between any two randomly chosen Ashkenazi individuals is roughly equivalent to what you would find between fourth or fifth cousins in most other populations. There's a genome-wide homogeneity among Ashkenazi Jews that is visible in every analysis, a fingerprint so reliable and so specific that consumer genetics companies like 23andMe can identify Ashkenazi ancestry from a saliva sample with near-perfect accuracy. More accurately, in fact, than they can identify most other ancestries anywhere in the world. This homogeneity has direct and serious medical consequences. When a population passes through a severe bottleneck, certain mutations that happen to be present in the founding group get amplified, carried forward at far higher frequencies than they would achieve in a larger, more genetically diverse population. The founding individuals of the medieval Ashkenazi community happened to carry elevated frequencies of variants associated with several serious inherited conditions. Tay-Sachs disease, which destroys nerve cells in the brain and spinal cord. Gaucher disease, a lysosomal storage disorder affecting the liver and other organs. Canavan disease, a progressive neurological condition affecting infants. And variants of the BRCA1 and BRCA2 genes that substantially elevate the lifetime risk of breast and ovarian cancer. None of these conditions are unique to Ashkenazi Jews, but all of them occur at notably higher frequencies in this population than in the broader human gene pool. They are not marks of weakness or genetic inferiority. They are the biological footprints of a community that was repeatedly pushed to the edge of extinction, that survived massacres, pogroms, forced conversions, and ultimately the industrialized genocide of the Holocaust, and that still exists today, still passes those ancient variants from parent to child, still carries within its genome the molecular evidence of everything it has endured. The question of where the Ashkenazi people originally came from genetically, beyond the broad answer of the Middle East and Europe, has been the subject of sustained scientific debate. The most prominent alternative theory was the so-called Khazar hypothesis, which proposed that Ashkenazi Jews descend primarily not from ancient Levantine populations, but from the Khazars, a Turkic-speaking people of the Caucasus region who converted to Judaism sometime in the 8th or 9th century. The hypothesis attracted significant attention, partly because of its political implications for debates about identity and historical land claims in the modern Middle East. It was tested with the largest and most comprehensive dataset ever assembled for the purpose. Genome-wide data from over 1,700 individuals drawn from 106 Jewish and non-Jewish populations, including extensive samples from the Caucasus region that had never previously been included in such an analysis. The result was unambiguous. Ashkenazi Jews showed no particular genetic affinity with Caucasian populations. The Khazar hypothesis did not simply fail to receive support, it failed comprehensively across every analytical method applied to the data. What the data showed instead was a population with a solid Middle Eastern genetic core, consistent with origin among the ancient inhabitants of the Levant, combined with a meaningful European admixture component, acquired during the centuries of diaspora settlement. When Orchestra and earlier local ancestry inference tools examined which parts of the Ashkenazi genome carry European ancestry and which carry Middle Eastern ancestry, they found that the European contribution came disproportionately from Southern European populations, Italian and Iberian sources primarily, rather than from the Northern and Eastern European populations, among whom Ashkenazi Jews most recently lived. This tells us that the most significant genetic mixing with European populations occurred earlier in the diaspora, during the centuries when Jewish communities were concentrated in the Mediterranean world, before the major northward and eastward migration into the Germanic and Slavic lands that gave rise to the specifically Ashkenazi community as history knows it. The maternal line tells an equally revealing story. Mitochondrial DNA, inherited exclusively from the mother and therefore tracking female ancestry without the blurring effect of recombination, has been studied separately from the nuclear genome. A 2025 analysis found that fewer than 15% of current Ashkenazi Jews carry mitochondrial DNA that traces to European host populations. The vast majority of maternal data, which independently confirms Near Eastern origins on the paternal side as well, both lines of inheritance converge on the same conclusion. The Ashkenazi people are, at their genetic core, a Middle Eastern population that spent approximately 1,000 years in Europe, absorbed meaningful European genetic material during that long residence, primarily from southern sources and concentrated in the earlier phases, and emerged from that sojourn as something entirely distinct from both their geographic origin and their place of long residence. The broader lesson that this research teaches extends far beyond the Ashkenazi case. What the latest generation of genetic tools has demonstrated, with unprecedented clarity and at unprecedented scale, is that the concept of fixed biological race is not just inadequate as a scientific category. It is actively misleading. The standard continental labels capture a real statistical signal in the eye, from a very long film of freeze-frame data. But they describe a single frame pulled, extracted from a story that has been moving and changing for 200,000 years. They tell you roughly where your ancestors were at a particular window in time. They cannot tell you who those ancestors were, what journeys they had taken before arriving there, or what other populations they had merged with or given rise to along the way. A large American study published in Science in June 2025, one of the most expansive genomic analyses of the United States population ever attempted, found striking and systematic mismatches between the racial categories people use to identify themselves and the ancestry actually revealed by their DNA. Among individuals who declined to self-report any racial category, more than 90% identified as Hispanic or Latino, and their genomes revealed a broad mixture of African, European, and Native American ancestries in varying proportions, a genetic mosaic that resists any single continental label. The researchers noted that their database was more genetically diverse than even the dedicated global reference panels previously used as the gold standard for population genetics research. Nearly every major human population on Earth was represented among the participants. Even Japan, long considered among the most genetically homogeneous populations anywhere on Earth, turned out to carry a hidden third layer of ancestry invisible to earlier methods. A 2024 study from Riken overturned the standard two-ancestor model of Japanese origins, the Jomon hunter, and the continental rice farming migrants, by identifying a third contributing ancestral group with ties to Northeastern Asia, the historical Emishi people. Japan's internal genetic diversity is real, structured according to geography, and was simply invisible at the resolution of earlier analytical tools. The same pattern almost certainly holds for dozens of other populations we have not yet examined with sufficient care. And the institutional bias that shaped decades of genetics research compounded all of this. Most genomic studies historically focused disproportionately on people of European ancestry, a long-standing imbalance that shaped which disease variants were cataloged, which populations could benefit from precision medicine, and which human stories were being told. A 2024 Johns Hopkins study addressed this directly, generating a new global catalog of human gene expression data that included populations from around the world that had been poorly represented in the literature, building, at last, a view of human genetic diversity broad enough to actually serve all of humanity. What all of this means, taken together, is that the human story is not a story of separate streams flowing in isolation from a common source. It is a story of rivers, joining, diverging, joining again, carrying the sediment of every watershed they have passed through. Every population is a palimpsest, written over by migration after migration. Every genome is a document of movement, a record of survival. Every individual carries within them the traces of people who lived and walked and crossed seas and endured disasters across tens of thousands of years, across ice ages and continental migrations we are only now beginning to fully reconstruct. The Ashkenazi case is the sharpest illustration of this principle precisely because it is the most extreme example available to us. A community that maintained its genetic cohesion through centuries of persecution survived a medieval bottleneck so severe it reduced the founding gene pool to a few hundred individuals and emerged from that compression carrying a uniquely concentrated genetic signature that now gives science one of the clearest windows we have ever had into the mechanics of how history shapes biology at the molecular level. Their DNA is unusual because their history is unusual, not because they stand outside the human story, but because they demonstrate with extraordinary clarity what is true of every population on Earth, that who we are in our chromosomes is inseparable from what we have been through as a people. When scientists say that one group's DNA does not fit the standard picture, what they're really saying is that the picture was never large enough to hold reality. The Ashkenazi Jewish genome does not fail to fit human history. It fits human history perfectly and completely. It simply refuses to be flattened into a label, refuses to be reduced to a continental category, refuses to be explained by a single snapshot taken at a single moment in an enormously long and complicated story. And in that refusal, it speaks for every human genome that has ever been sequenced. including yours, because none of us are simple. None of us arrived at this moment without carrying the marks of every journey, every merger, every survival that preceded us by thousands of years. The science of DNA is not the science of difference. At its deepest level, it is the science of connection, the slow, painstaking, humbling reconstruction of a single long story that began in Africa, moved across the entire surface of the earth, branched and wove and collapsed back on itself in ways we are still learning to read, and continues today in every person breathing. The map is finally being drawn, and every one of us is on it.